Section 5.3 is on active transport across the membrane. So passive transport did not require extra energy because it went with the concentration gradient. Active transport requires it because it actually goes against the concentration gradient. Because it goes against the concentration gradient, it's like being in a boat and trying to paddle upstream. It goes from low to high concentration, so it does require extra energy. That energy is in the form of ATP, adenosine triphosphate. That's how all of the energy is used inside your body, is in that form. So an example of this, we talked about this before, but it's a sodium-potassium pump. This uses ATP to move sodium ions out of the cells and the potassium ions into the cells, and that's against the concentration gradient. It's against the regular flow. The change of shape in the active transport carrier protein, it has to happen because it's going to bind to the sodium ions and to the potassium ions, and they're not identical. So let's take a look and see what that would look like. Here we have the carrier has specific shapes for sodium and for potassium, and this is inside the cell and outside the cell. For every 3 sodium, it can bring in 2 potassium. So here we have ATP, and that's going to push that sodium out of the cell, and it's going to turn around and take potassium into the cell. Macromolecules are transported into or out of the cell inside of vesicles. So this is considered bulk transport because it won't go through the membrane. It actually has to have the membrane surround it and either pull it into the cell or get rid of it out of the cell. So you need to know the difference between exo and endocytosis. Both of these are active transport because it takes energy, requires energy in the form of ATP. Exocytosis is when a vesicle fuses with the plasma membrane and secretes its contents. Exo is out of the cell, so taking something out of the cell and releasing it. Endocytosis is bringing it into the cell, and the cell will engulf the substance into a pouch, and that becomes a vesicle. There are different types of endocytosis. We have phagocytosis, which is where we have solid materials taken in. Pinocytosis is where we have liquids or very small particles taken in. Then we have receptor-mediated endocytosis. In this type of endocytosis, it's actually a specific pinocytosis that uses a receptor protein and a coated pit in order to send out a signal for a specific vesicle to be formed. The endocytosis and the exocytosis, if you look at it this way, exocytosis, well, let's talk about endocytosis first. You eat and you drink separately. So phagocytosis is solid, pinocytosis is drinking, so we do these things independently of each other. You eat and you drink. However, when you throw something up, it all goes out at the same time. Exocytosis is like that, so there's not a specific solid or specific liquid that's in it when it is taken outside of the cell. This is just showing exocytosis, where you have a secretory vesicle, and it's going to merge with the plasma membrane and be taken outside of the cell. Phagocytosis is taking in a solid. Your white blood cells do that when they take in bacteria and render them harmless. Pinocytosis is when you have small particles or liquids. Then we have the receptor-mediated, where you have coated pits, and they are specific for a particular solute to take inside the cell. That's going to finish up this section. It's a really short section of notes. What we have to remember about passive and active transport is that passive transport does not require ATP. It goes with the concentration gradient. We talked about examples of that of diffusion, facilitated diffusion, and osmosis. Then we have active transport that does require ATP because the substances go against the concentration gradient. So it's like paddling upstream, requiring energy. And the examples of that were the sodium-potassium pump, and then we have endo and exocytosis as well.